Great, take it away.
Super duper. Thanks, Manolis. So I'm delighted to welcome Manolis today for a Computational Health Center seminar. I don't think Manolis requires an introduction. Nevertheless, he's a professor of computer science at MIT, member of the Broad Institute, principal investigator of the Computer Science and Artificial Intelligence Lab at MIT, and head of the MIT Computational Biology Group. Now, Manolis has received a very, very long list of awards, and I can't really go through all of those, but we are so grateful to have you here today, Manolis, and we really look forward to your talk. So without further ado, I'll let you tell us all about your exciting science.
Wonderful. Thank you so much, Ellie, for the very kind invitation. Thank you, Fabian, for being just an awesome co-host here and always welcoming me in Munich. I had a wonderful time last time, and I'm coming back. So I'll be there in person.
Yeah, 2024. We're
Yes.
looking forward to that.
I look forward to that.
And I'm sure you're going to bring a drone, huh?
Oh, gosh. My latest drone has goggles, so it's like a whole other level.
I showed this to my kids, but I follow up on this later because he built one of these. Sorry, Eden.
Okay, great. So let me just stop and reproject. For some reason, it doesn't hide the bottom here, so I'm going to move it there. There we go. Okay, great. So so it's a real pleasure to be here. And what I'd love to tell you about is our work on really understanding and manipulating disease circuitry at single cell resolution. So how do we translate genetics and genomics into mechanism and ultimately therapeutics? So I'm a member of the MIT, of the Computer Science Lab, and of the Broad Institute, and we work basically on genomics for human disease. The basis of our work starts with genetics, and genetics is not a new thing. For thousands of years, humans have selectively bred animals for their properties, crops, to go from teosante here, this is something completely unedible, to corn that basically feeds a whole continent, from something that would eat you to man's best friend. So we've understood the properties of inheritance and selection for a long time across the plant and animal kingdom, but also for humans. The inheritance of eye and hair color and height have very long been understood. But it's not until 1860 or so that Gregor Mendel, through a series of very precise experiments in peas in Brno in Europe, basically figured out the rules of inheritance, that inheritance is discrete, it's not blending. And there's dominant recessive alleles that can jump through generations, and you have multiple loci that we now call the genes that are independently inherited, creating the combination of phenotypes that we see across the natural kingdom. But Gregor Mendel's views basically remained fringe for more than 50 years because people really didn't pay attention. They couldn't see how to reconcile these beautiful, elegant experiments in peas with the seemingly continuous variation in humans. Eyes do not come just in two colors. There's a continuum of colors, a continuum of hair. There's a continuum of height. There's a continuum of every human trait. And this was very difficult to reconcile. And it's not until the beginning of the 1900s that Ronald Fisher and other statisticians basically realized that, wait a minute, the two do not have to be continuous. It's not just one gene controlling human height. It's maybe dozens, maybe hundreds. And if you look at the additive effect of even a small number of loci, such as 5, you could still get a seemingly continuous distribution of height. So you could explain the two models together simply through additive effects of independent genes contributing to the same phenotype. And that poses a challenge because the classical, quote-unquote, Mendelian traits could be mapped simply by the deviations of Mendel's laws of inheritance, namely the deviation from independent assortment could lead to some kind of co-inheritance of different loci, different traits relative to each other, and therefore paint where they are on the chromosome. But for complex traits, this is very different because for complex traits, we basically have thousands of alleles scattered across the chromosomes, all contributing a minute amount to human disease. So it's not until the sequencing of the whole human genome, the building of variation maps and haplotypes, and the power of genome-wide association studies that we could finally start mapping complex traits. So that's where we are today with hundreds of thousands of genetic variants that we know play a causal role on disease. And the causal part is extremely important because it basically says that we can actually understand mechanism through this causality. And that's sort of what my talk will be about. So basically, how do we now take these, incredible discoveries of genetic associations and translate them into mechanism? So we basically want to deliver on the promise of genetics. And what is the promise of genetics? The promise of genetics is that if you look at something like obesity that has traditionally been simply blamed on the individual, that, oh, you're obese because it's your choice to exercise or not, diet and so on and so forth. When we look at body mass index, we basically see that there are indeed across the 23 pairs of human chromosomes, many loci that are hugely significant. In this particular case, the FTO locus, which has been since renamed fat and obesity associated, is basically reigning supreme among all of them. And what that basically means is that there's something in this region going on that contributes to obesity. So that gives us promise that we can intervene therapeutically, that we can actually have a path to understanding causality, because throughout human history and throughout the history of epidemiology, it's all been about correlation, not causation. But with genetics, you basically have temporality for free, because these variants were inherited before I developed obesity, and therefore it's much more likely that these play a causal role rather than my inheritance patterns are a consequence of my obesity. We can also start understanding mechanisms. The power of genetics is that it doesn't matter how something functions, you can still discover these loci. And if you go into those loci, you can understand new mechanisms, develop new target genes, new therapeutics, and enable precision medicine and personalized medicine. The challenge is that for 93% of these loci, it's very difficult to get at mechanism. Why is that? Because the vast majority of them are in fact non-coding. 93% of cases, you don't see a single protein coding alteration. In this particular case for FTO, you basically see that there are 89 common variants that are co-inherited, all hugely significant between these two haplotype recombination blocks. And you basically have this whole region that could be causal. You don't know which one it is. Moreover, the exons are these tiny little dots, exon 2, exon 3, and then you have these giant introns that basically are associated with the disease, but we don't know how they act. So that's the challenge of mechanism. The power of genetics is that it doesn't matter how something functions, you can discover it. The curse of genetics is that when you discover it, you still have no idea how it functions. And that's where we come in. We basically say, we need to understand the target gene. We need to understand the causal variant. We need to understand the cell type of action, the relevant pathway, and the mechanisms. And in this particular case, the FTO gene itself turns out to be a red herring. In this particular case, the regulatory control regions we showed in our own work published in the New England Journal of Medicine, we showed that it is, in fact, two different target genes that are sitting 1.2 million nucleotides away and 600,000 nucleotides away from this region that are, in fact, the true driver genes. So I was introduced one time as the author of one of the most depressing papers of that year, because for many loci, when there's a single gene, people are very happy. They're like, aha, we know the gene. But what we showed is that even though you have a single gene that's kind of far away from all the other nearby genes, the true targets could yet still be something completely different. So how do we do this? How do we find those things systematically? How do we understand target gene, causal variant, cell type of action, relevant pathways? The way we do that is that we start with genetics. Here I'm showing you a different example for ApoE. This is the strongest genetic association with Alzheimer's disease, and specifically the ApoE4 allele versus the ApoE3 allele. And we start with disease genetics to reveal both common and rare variants or regions that are playing causal roles. We then want to know how do they act, and we profile systematically the molecular impact of those variants to get at the genetics, to get at the causality, at the mechanism. We basically profile RNA, which is transcription, the quote-unquote output of that region, to see how ultimately gene expression is changed, but also the circuitry through the epigenome. So we look at DNA accessibility, histone modifications, nucleosome positioning, DNA methylation, that basically allows us to say which regulatory control regions are active in every cell type and how do they vary between individuals to understand the impact of, number one, the genetic variants by looking at variation in healthy individuals, but number two, the impact of disease itself, some of which could be environmental, some of which could be a consequence of long-term dysregulation that ultimately changes between healthy and control, between disease and control samples. So we then gather all these data sets across sometimes thousands of individuals, and we integrate all this data to predict what are the driver genes, regions, and cell types that are responsible for the phenotypes observed. We then use, these deep learning methods, these Bayesian inference methods, these causality identification methods, and then we predict these drivers, and then we go and manipulate these drivers. So we can use human cell cultures to alter these genes or control regions in specific cell types, or we can use animal models to basically look at the organismal effect of these predictions, and of course, disseminate the results and start the cycle all over again. So what do I mean by circuitry? By circuitry, I mean, we look at not just, hey, there's something going on here within the last introns of this gene, but instead, here are the specific mutations. These are the specific genetic differences between risk and non-risk individuals. It's not just some abstract concept. These are the SNPs. And these SNPs are overlapping very precise enhancer regions. These are the long-range acting control elements. And then these enhancers are active in very specific tissues and cell types. Here's the motifs within them that are enriched within these tissues and cell types. And here's the regulators that are recognizing these motifs, giving us a circuitry. And then lastly, once we have that regulatory control region, here are the candidate target genes for that control region, which we can find out through chromatin conformation capture to look at the folding of the genome in 3D. You can look at correlation analysis between the accessibility of these regions and the expression of these genes. And you can look at also genetic QTLs, so expression quantitative trait loci, that basically look at how genetic variation in these SNP is in fact correlated with expression variation in these genes. So using these three lines of evidence for linking, we can now go from SNPs to regions, to target genes, to cell types, regulatory motifs, upstream regulators, and ultimately pathways of function. So in the case of the FTO locus that I showed you earlier, we wrote a whole paper about a single bit of information in the human genome. We basically talked about this T to C alteration. So we were able to fine map 89 common variants in this locus that I showed you earlier into a single letter change that is disrupting an AT-rich motif, causing loss of binding of this ARID5B repressor. When the motif is lost in the C individuals, which are obesity risk, the regulator can no longer bind. It can no longer repress this super enhancer, which is 12,000 nucleotides within the first block that I showed you here. Basically, the causal variant we showed is RS1421085. And then there's a 10 kb super enhancer right in that element that then targets these two genes at 1.2 million nucleotides distance for RX5 and 600,000 nucleotides for RX3, several genes over. Now, we then need to figure out what do these genes actually do. And through a series of experiments, both computational and experimental, we basically showed that these are in fact previously undiscovered master regulators of thermogenesis. If you look at a thermodynamic equation of energy transfer through our food to our exercise, we can basically see how many calories do we consume, how many calories do we burn by moving around or thinking, and how many calories are we left with at the end of the day. We can now choose to store these calories in this incredible process of generating lipids and creating this gradient in the mitochondrial membrane that basically generates ATP and stores it in these lipid-rich molecules that basically then turn white adipocytes into these big, juicy energy storage machines. That is truly a miracle that you can basically eat on Monday and still have calories left on Friday by storing them. But this was great way back when. So it rose from 2% frequency to 40% frequency in Europe. So European chromosomes basically have 40% obesity association, whereas African chromosomes only have 2%, suggesting that perhaps it's not just drift, but it might actually be selection going through the deserts, going through ice ages, and basically being able to store calories. But a subset of our adipocytes can basically choose to instead burn these calories, which again is nowadays very beneficial. So you can basically depolarize the mitochondrial membrane, cause a loss of the proton gradient, and instead lose calories through heat. So again, in a thermodynamic equation, you have energy storage, energy utilization, and then energy loss through heat. So this is a regulated process, and master regulators turn out to be IRX3 and RX5. And downstream of them, we have uncoupling protein 1, PGC1 alpha, etc., that basically have multiple ways of burning calories. Why are we excited about this circuitry? It's nice to know why, we are obese or lean. So basically, in my particular case, I'm actually homozygous risk for the obese allele. So thank you both, mom and dad. They both gave me a non-functional C letter here. And therefore, my ARID5B protein cannot bind in my motif. So I kind of have to watch my diet and exercise in order to avoid being overweight. So basically, that locus alone provides 7 additional pounds of weight to an adult individual. And again, compounded with polygenic risk, you can basically have a predisposition to obesity. But why are we excited? Not just because we want to tell people, sorry, bad luck. We want to now actually intervene. And that's why it's exciting to uncover and manipulate the circuitry, because we can reverse the disease phenotypes. So we can increase or decrease the expression of the upstream regulator and switch between lean and obese phenotypes. We can increase or decrease the expression of the downstream target genes, and again, switch between lean and obese phenotypes. And we can also go and edit with CRISPR-Cas9 genome editing the single letter from C in risk individuals to T, and then restore the thermogenic operation. So as we showed in the series of elegant experiments carried out in collaboration with Hans Kauner's lab and led by Melina Klausnitzer, we basically were able to show that risk individuals like myself, who are unable to thermogenize, here measured by oxygen consumption rate, we can basically change the single letter and restore both basal and isoproterenol stimulated thermogenesis. And this is very exciting. It basically says that when it comes to precision medicine, changing 1 letter out of 3 billion letters in the human genome can basically restore a cellular phenotype by a factor of 7, which is quite remarkable. But then we didn't stop there. We also went to mice in collaboration with the University of Toronto, and we basically showed that under control of an adipose promoter with a dominant negative construct for IRX3, you can basically lead to dramatic weight loss for these mice. And most importantly, when you put them on a high-fat diet, regular mice gain weight, and these IRX3 dominant negative mice, where this regulator is repressed, are simply unable to gain weight. They cannot store white fat. If you open up these mice, you basically see these very red organs. It's all brown fat, which is quite remarkable. So we start with a relatively weak effect. 7 pounds is still quite dramatic for human genetics, but it doesn't matter how big the effect is in the human genetics. What's really exciting is that we get the circuitry and the causality, and then we can intervene by turning these knobs a lot more than what natural selection will tolerate in the human population, and therefore have these dramatic therapeutic effects. So it doesn't matter how you gain weight. By altering your metabolism, we can make sure that you burn it. And that's why we're so excited. So this is one example. We have many additional examples. Just a few weeks ago, we basically published in Nature another story looking at the strongest genetic association now with Alzheimer's disease. So I showed you this picture earlier. ApoE4 confers a 10-fold increased risk for Alzheimer's disease in risk individuals, in homozygous risk individuals. So we basically looked at male and female individuals, both ApoE3 and ApoE4, and both with AD pathology and without AD pathology. And we basically asked, what is the effect of the ApoE4 allele at the molecular level? So in this particular case, we carried out single cell RNA sequencing, and then we looked at all of the cell types between excitatory neurons, inhibitory, astrocytes, microglia, oligodendrocytes, and oligodendrocyte progenitor cells. And what we found is that one of the most altered pathway was in fact cholesterol biosynthesis, and specifically in oligodendrocytes. And through a series of elegant experiments, again, led by Joel Blanchard in collaboration with Li Guitai's lab, we basically showed that what this cholesterol biosynthesis dysregulation is about is cholesterol transport, exactly as you would expect for an apolipoprotein E, cholesterol transport. So what we showed is that cholesterol transport is disrupted specifically in oligodendrocytes, the cells that are responsible for forming the myelin that protects the neurons. And what we showed is that if you are homozygous risk for APOE4, with or without Alzheimer's disease, you basically have this accumulation of cholesterol in the endoplasmic reticulum of your oligodendrocytes. And the APOE3 individuals have these beautiful myelin sheath surrounding the neurons. And the APOE4 individuals, you can see many neurons have absolutely no protection. And generally, it is much reduced. So we basically said, what's going on? Is this a consequence of Alzheimer's, or is this a cell autonomous effect? And we were able to go and show that even in an iPSC-derived line that is obviously not suffering from environmental consequences, you see this dramatic change in lack of myelination. And even in a mouse model, you see this dramatic change. And what's really very exciting is that by restoring cholesterol transport through cyclodextrin, we can restore, number one, myelination, because the cholesterol is no longer stuck in the endoplasmic reticulum. It can form these myelin protection. And you also restore cognition in a mouse. Again, another example, but we want to do this systematically across hundreds, thousands of these genetic loci. So how do we do this systematically? We basically need to, number one, build reference epigenomes to understand what are the disease-relevant tissues and circuits. We also need to build reference maps of regulatory control elements through comparative genomics. We then need to understand how these maps vary from person to person, how they act at the single cell level, how they're influenced by genetic effects, how they act across multiple phenotypes in multiple tissues, and then how we can systematically manipulate them. So let's dive right in. How do we understand how these genetic variants are acting and which tissue they're acting in? So we basically have built these reference maps of the human epigenome in collaboration with the Roadmap Epigenomics Project, ENCODE, and more recently, the IHEC, International Human Epigenome Consortium. We've looked across adult, embryonic, different brain regions, iPSCs and iPSC-derived cells, primary cells across different places in the body. And for each of those, we looked at promoter-associated histomodifications, enhancer, transcription, polycomb, heterochromatin, and generally activation-associated histomodifications, DNA accessibility, DNA methylation, and gene expression. We then build techniques for learning the quote-unquote language of chromatin by discovering systematic patterns of combinations of these marks across the genome. And then we assigned names to these combinations based on their functional enrichments and called some of them enhancers, promoters, transcribed, repressed, and so on and so forth, through a series of papers you can see here. So every one of those is a QR code. You can just scan it and go to the corresponding papers. We can then take these enhancer regions that vary dramatically from cell type to cell type and see how are these enhancer annotations active in different cell types, correlated and overlapping with genetic variants scattered also across the genome and associated with different traits. We can look at cholesterol here, for example, and we can see that that liver enhancers are overlapping very strongly with it. We can look at height-associated genetic variants, and they overlap enhancers active in embryonic stem cells. These immune-associated genetic variants overlap enhancers active in T cells, B cells. If you look at blood pressure-associated genetic variants, you see that they overlap preferentially enhancers active in the left ventricle of the heart. If you look at fasting glucose-related traits, you see very precisely pancreatic islets, very consistent with the biology of type 2 diabetes, for example. If you look at inflammatory bowel disease, you can see immune cells and also digestive tissues very strongly enriched, just like you would expect for inflammatory bowel disease. But surprisingly, for Alzheimer's, we saw no enrichment for brain. Instead, we saw a very specific enrichment for CD14 plus monocytes. These are the circulating monocytes, but the CD14 plus mark is also there for these tissue resident macrophages, which are resident in many tissues, but also the microglia of the brain. So the specialized macrophages, specialized immune cells of the brain. So that basically made us reason that perhaps the majority of genetic risk across Alzheimer's might in fact be acting in a inflammatory and immune pathway instead of the neurons directly. And indeed, when we did cell sorting experiments of neurons versus microglia versus other glia, we found that very specifically the LD score regression enrichment of genetic variants associated with Alzheimer's was localizing in enhancers active in microglia through cell type specific H3K27 acetylation through these pull down experiments. And indeed, we were able to show that if you look at the progression of Alzheimer's in a mouse model developed by Li Hui Tsai in her lab called CKP25, that shows both early and late stage symptoms of AD, across time, we found that immune processes alter very early on, but neuronal processes alter only later on. That suggested that perhaps inflammation might be a causal component of Alzheimer's disease rather than a consequence of neuronal changes. We have since scaled up from 127 epigenomes in the Roadmap project to 834 to now more than 1,000 epigenomes through the IHEG project, and that increased the number of enriched traits from 54 to more than 200. It allows us to paint much more complex pictures of where are specific genetically associated variants with diverse traits enriched. And for example, you can see here educational attainment, intelligence, cognition, localizing very specifically in the brain. That's where excitatory neurons are the most abundant, but also schizophrenia, neuroticism, worry, depression, and so on and so forth. You can see conversely that Alzheimer's localizes very specifically in immune cells, as we saw earlier. You can see that cholesterol localizes in liver, heart disease with heart, and so on and so forth, filtration with kidney. But you also see these traits in the middle here, and every single one of these circles is a pie chart that tells you the diversity of tissues that are involved and implicated. And specifically for coronary artery disease, one of the most polyfactorial traits, we basically said, hey, can we start partitioning the complexity of these different tissues that are implicated through this enrichment into specific biological processes that are associated with the subset of variants that are associated with different tissues. If you look at QT interval, educational attainment, you see very specifically heart or brain. If you look at Alzheimer's, you see both immune and brain. If you look at body mass index, you start seeing a larger diversity of muscle, adipose, kidney, and so on and so forth. If you look at health span, you see many, many additional tissues. And if you look at coronary artery disease, you see liver, coronary artery, thyroid gland, adipose, gastrointestinal, and so on and so forth. But if you look at the subset of variants that are associated with liver enhancers, you basically can see that they play very different biological roles than the variants associated with CAD that are instead localized in coronary artery enhancers and thyroid enhancers, and so on and so forth. So that's very exciting. It basically means that we can partition complex traits into their parts and start thinking about the hallmarks of these different diseases. And that applies to individual loci. You can basically go through the most strongly associated variants and then start asking what is their overlap with the most strongly enriched tissues. And what you can see is that some variants are basically very specifically coronary artery, others are very specifically liver, and others are sort of quite broad, overlapping enhancers across all of them. And you can now take these examples, such as PCSK9, and say, okay, what does our analysis tell us about the tissue of action and the target genes? So if you go to compbio.mit.edu/epimap, you can find browsers of 30,000 of these loci. And for every one of those, we basically show the strongest enriched, the most strongly enriched tissues, the genetic variants, the linking between these genetic variants and downstream target genes. And then in these examples, you can see that in some cases, you see very specifically liver associations. In other examples, you see very specifically hard associations with eDNRA, example number 2 here. And in other examples, you see both. For example, you see these genetic variants here that are linked through both liver and coronary artery, targeting different genes for different links. This is very exciting. Basically says that perhaps this locus rose to genome-wide significance because it doesn't just act in one, but maybe in 2 different tissues. And in this particular case, maybe through multiple genes. So this is just one example of such pleiotropy in the human genome, which appears to be very much the rule rather than the exception. So this is all building these reference epigenomes through these histomodification marked DNA accessibility, but we can also build these reference functional maps through comparative genomics. And in particular, we have developed a series of evolutionary signatures for discovering protein coding versus non-coding genes, RNA structures, microRNAs, regulatory motifs enriched across the genome, and individual motif instances through a series of papers through the years. And we can use those to start now asking, how are these annotations overlapping with the biological functions that we see in these genetic variants? One approach is to basically look for new protein coding exons. And when we did that in collaboration with GenCode through a series of papers, we basically showed that indeed we can discover more than 100 new exons that were previously undiscovered, and that 118 of them are in fact overlapping genetic variants that were previously thought to be non-coding. But we were simply blind to the exons that are sitting in these regions. In one such example, myopia-associated variants are in fact localizing very specifically in this retina exon that was previously undiscovered, but we now find to be expressed in a very cell type specific way, suggesting that perhaps this might actually be the causal variant in this region that was previously thought to be non-coding. And we have the same thing for many, many other loci. But we can also look at annotations of individual regulatory motifs and how they are disrupted by these variants. And we can use these techniques to also understand the SARS-CoV-2 genome underlying the COVID-19 pandemic, discover that perhaps ORF10 is in fact not protein coding at all, is instead a non-coding RNA. And then ORF3C is a new protein coding gene that we discovered overlapping these previously annotated ORF3A, but in a different reading frame through these evolutionary signatures. And we can also study how individual variants associated with increased virulence are in fact impacting proteins, and how the evolutionary speed across the sarbecovirus lineage in fact relates with the evolutionary speed in the current pandemic of individual genes. But everything I've showed you so far has been about these reference annotations. We can also look at the variation in these epigenomic and transcriptional impact to basically start bridging the gap between genetic variants and disease, and start asking what tissue, what regulatory control region, what gene is affected in the path of causality to disease. But the challenge here is that some of these are in fact downstream of the disease state, and some of them are in fact arrows flowing in the opposite direction, or both influenced by the environment. And in particular, if you look at a gene that increases with expression correlated with Alzheimer's disease, you might want to say, well, maybe this gene is actually causing disease, or maybe this gene is because of the disease, or maybe this gene is actually combating disease. So you don't want to repress this gene necessarily, but you want to sort of understand the causality in that path. And the way to do that is look at mediation analysis, namely, what are the mediators of the genetic effects all the way to phenotype? In particular, we can first look at this arrow and discover genetic variants that are predictive of, say, DNA methylation in this example. And we find 50,000 such significant QTLs in the human genome, where if I know your genotype at birth, I can basically predict your methylome in the brain at 93 years of age. And we can basically do that systematically and start inferring methylation. Instead of just looking at genome-wide association studies all the way here, we can look at methylome QTLs and start predicting methylation. We can also look at methylome-wide association studies to look at this bidirectional arrow of the correlation between methylation and disease. But here we need to have access to many, many such epigenomes. But instead, if I can build a predictive map in the subset of individuals that we have methylation for, I can start predicting the methylation of new individuals across a very large cohort, and then start correlating the predicted methylation from the genetics with overall disease status. And that allows us to now start discovering new genetic variants that might be causally contributing to Alzheimer's, specifically mediated through epigenomic effects. And we can trace that path of causality through genetics, methylation, transcription, and disease, and then discover new genes that are now in existing loci that I'm showing here in purple, or new genes in previously non-genome-wide significant loci in gray that we can now turn into genome-wide significant because we are testing many fewer hypotheses. We're only testing transcriptional associations rather than every SNP in the genome. So all of this has been now at the tissue level. Let's now dive at the single cell level. The example that I showed you with microglia in the brain basically tells us that sometimes tissue level data might be hiding effects that are actually happening in less abundant cell types. So to do this, we basically started profiling at single cell resolution hundreds of individuals. And we have written a series of grants that have now allowed us to profile Alzheimer's disease, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis, schizophrenia, bipolar disease, Down syndrome, autism, PTSD, addiction, aging, cardiac disease, obesity, and exercise across healthy and control and disease individuals, across different genetic carriers, and across both men and women, both young and old. And we're looking at dozens of cell types for each of those, tens of millions of cells, multiple brain regions very often for the brain-associated disorders, and looking at both DNA accessibility and RNA expression, and increasingly looking at multi-omic data. So in the first paper that we published a couple of years ago, we basically found that individuals with Alzheimer's disease show dramatically altered transcriptional pathways across every major cell type, not just in microglia, as we expect the genetic effects, but across excitatory neurons, inhibitory neurons, astrocytes, oligodendrocytes. We could actually find subsets of these cells that were associated with high or low amyloid, with late Braak stages or early Braak stages, with high cognitive decline or low, with AD or non-AD. And quite surprisingly, many of those correlations also correlated with the sex of the individuals. Male individuals showed many more quote-unquote healthy cells, and female individuals showed many more quote-unquote AD-associated cells, suggesting in fact that there's a lot more transcriptional resilience perhaps in the women. And there's a lot more transcriptional burden anyhow. And again, this is of course associated with female individuals showing increased Alzheimer's burden, even when you correct for the longer longevity of women. We also found that early stage changes in AD were very cell type specific, but late stage were in fact much more shared between tissues and cell types. We found that AD increased the number of differences we found between male and female individuals. So there's 3,000 genes that are differentially expressed in excitatory neurons prior to AD, but 6,000 after AD, suggesting that we should be really thinking about AD-specific and sex-specific alterations. And in particular, we found that myelination differences were quite dramatic between men and women. And indeed, we predicted that we would see more white matter loss in the female individuals that basically showed lower activity of myelination pathways. And indeed, this is what we saw, and we confirmed a finding that was initially purely transcriptional. We have since expanded from 80,000 cells in our first paper to 1.6 million cells in a paper that is currently in revisions. And we basically are able to annotate not just excitatory inhibitory neurons, but dozens of subtypes of excitatory inhibitory neurons. Here we're looking across prefrontal cortex, midtemporal cortex, angular gyrus, entorhinal cortex, thalamus, hippocampus, and mammillary body. And we can distinguish 30 different subtypes of excitatory neurons and 23 different subtypes of inhibitory neurons. And these vary dramatically between brain regions. So for example, you can see these inhibitory neurons for anterior thalamus are quite distinct from the ones that you see in other cell types. And then similarly, you see that even glial cells, such as astrocytes, vary dramatically between mammillary body or thalamus or subcortical regions and neocortical regions. We can use these 1.6 million cells to now sort every single cell according to how AD-like is its transcriptome. And what we see is that indeed we can distinguish early versus late acting genes based on their expression and their differential expression in this time course, and also genes that appear to be acting in the middle of the course of Alzheimer's progression. And we can use that to start perhaps even suggesting what treatments we should be targeting at different ends of this transcriptional time course. Using the anatomical sub-regions of hippocampus between dentate gyrus, CA123, subiculum, and the different layers of the entorhinal cortex, we can basically see that transcriptional clustering, in fact, recapitulates the anatomical position, and we can in fact distinguish specific anatomical regions that are more or less predictive of Alzheimer's disease. And we can use this to now start connecting the three-dimensional position of individual cells through reference data sets by building this deep learning model that allows us to predict the localization of both neuronal cells, but also glial cells across different layers, for example, of the cortex. We can also use the convolution of spatial transcriptomics to infer changes that are happening at single cell level, in this particular case for Purkinje cells in SCA1, spinal cerebellar ataxia, SCA1. And then what we can see is that the marker gene for Purkinje cells appears to be unchanged, but the moment we do the convolution, we can actually see quite a dramatic change of these cells themselves. We have expanded to subcellular resolution spatial transcriptomics. In this example, I'm basically showing you coronary artery disease across myocardial infarction, heart failure, ischemic heart disease in controlled individuals. And then we can define different landmarks of intima, lumen, or media, for example, in the artery, and then look at the differential expression of individual genes within individual cells of this. And we're writing a series of spatial algorithms for being able to define these landmarks and then recognize landmark-associated transcriptional changes and also correlations between cells. And what we're finding using those algorithms, we're finding changes that are very spatially localized. For example, for foam cells, these inflammatory changes that are happening in coronary artery disease are in fact appearing to be very specifically localized. And we can discover all of these networks. We're doing a lot of work now in translating between ATAC and RNA, basically building these joint representation models that are in some cases very specifically informed based on the attention weights that we're paying to specific peaks, enabling us as a byproduct to start linking peaks to their target genes. And then ultimately, we're hoping to translate also between spatial features using these kind of clip mixed architectures. And ultimately, we want to connect all of this to a diversity of data sets across omics, metabolomics, microbiome, EHR, imaging, and so on and so forth. Using these millions of cells, we can actually start partitioning the complexity into different modules and then discover these very coherently expressed across our millions of cells genes that are changing now as a group based on their common regulatory processes upstream of them. And that allows us to now go even into rare cell types like microglia or vasculature and look at the modules for how these are changing, which are much more robust than the expression of individual genes. We can also look at neuronal regions that are known to be connected through these long-range projections and see how they're changing transcriptionally. And what we're finding is that connected regions are in fact correlated in their transcriptional changes, enabling us to now start predicting what are the drivers of these inference, and then start inferring not just spatial organization, but also connectivity information across human and mouse. In the case of schizophrenia, we're able to now look at prediction of which individuals are schizophrenia-like in their transcriptome and control-like in their transcriptome. So just like we have PRS scores for polygenic risk scores, we have these polytranscriptomic scores, if you wish, that allows to now start predicting these individuals that might be at risk. And indeed, we find that schizophrenia individuals are much more to the right of this plot and control individuals are much more to the left. But there's some exceptions. So for example, this control male individual was not diagnosed with schizophrenia, but instead his son was actually diagnosed, suggesting that perhaps we can see something in the transcriptional profiles that might be missing from the diagnosis. And we see this systematically in bipolar disorder, in Alzheimer's disease, where there's a partition that happens from the transcriptome. And in some cases, we have these surprises. In schizophrenia, we basically showed that these surprises were sometimes associated with a distinct cellular state, which we call SZTR for schizophrenia transcriptional reversal, that allows us to perhaps start reasoning about either resilience pathways or alternative paths to schizophrenia. But more importantly, we can basically look at individual genes that are associated with individual genetic loci, and then see whether these genes are linked by high-C evidence, EQTL evidence, and so on and so forth, and also the direction of effect of these target genes for these loci and the cell type of action where they act. We can use this to start inferring upstream regulators and the modules with which they coordinately act. And we can group those into synaptic signaling, synaptic organization, and neurodevelopment. And specifically, these one module of coordinated regulators upstream of those appears to be particularly involved in early development and also in adult function. So this is psychosis in schizophrenia. We also looked at psychosis in AD. A subset of AD individuals basically show these psychotic episodes. And one would have thought naively that perhaps these are associated with increased expression of excitatory and inhibitory neurons, but we see exactly the opposite. That in fact, reduced expression appears to be the norm for these individuals that show psychotic episodes. And you see specifically enrichment for ribosome biogenesis, neuronal projection, autophagy, apoptosis, and signaling, among other pathways. In a paper that we just resubmitted to, we basically see these very similar results between ALS and FTD. So ALS is a motor disorder, FTD is a personality disorder. And yet what we're finding is that they appear to be affecting orthologous cell types in orthologous brain regions. In fact, what was quite surprising is that when we used motor cortex for ALS, we also profiled motor cortex as a control in FTD. And we used frontal cortex for FTD, and we also profiled frontal cortex as a control in ALS. But it turns out that both brain regions are affected in both diseases, suggesting that there's much more similarity than previously appreciated. And in fact, these von Economo neurons that are long-range projecting in the frontal cortex are orthologous transcriptionally to these BET cells, which are long-range projecting in the motor cortex, and they are the two most affected cells in both disorders. So this is consistent with the fact that individuals are sometimes diagnosed for one trait, but then the diagnosis changes post-mortem to show also the other trait, and that even within the same family, individuals that, for example, carry the C9 ORF72 mutation will manifest either as FTD in some individuals or as ALS in another individual, suggesting, in fact, a lot more overlap between these disorders. And we've now profiled disorders across the whole spectrum of PSP, CBD, behavior variant FTD, PPA, and so on and so forth. So we're very excited to start understanding this spectrum and understanding these relationships between them. We just had a paper in Neuron and Nature Communication with the Miriam Heyman lab and Anne Grabeel lab, looking at indirect pathway versus direct pathway spanning projection neurons and the seeming loss of identity and the specific loss in Huntington's disease. We're collaborating with Rudy Tanzi in pathogen-associated changes in AD, looking at specifically individuals with cytomegalovirus or simplex virus, and we're finding these very dramatic immune and vascular associated changes for those individuals. We also looked at COVID brain and found individuals with long COVID, and we saw that these individuals have an increased proportion of astrocytes and also dramatic dysregulation, not just in microglia pathways, but also in vascular pathways with angiogenesis, vascular development, and so on and so forth, as you would expect for a vascular disorder like COVID. And indeed, a lot of terms associated with RNA processing, but also downregulation of synaptic organization, neuronal projection, and generally brain function. I mentioned the story with ApoE4 and myelination at the introduction, but we also looked at a diversity of additional lipid transporters, such as ABCA7, looking at both common and rare variant carriers and how these are changing between individuals. NASA, in collaboration with Lihuitai, is basically leading this project to identify subtypes of microglia and how these are in fact associated with the ability to reprogram microglia in Alzheimer's disease, and basically go from homeostatic to inflammatory and back by overexpressing the regulators that are responsible for those, and in showing experimentally that we can indeed reprogram these inflammatory microglia to become more homeostatic, which is very exciting because that's thought to be one of the drivers underlying Alzheimer's disease. We had a paper looking at the vasculature in about exactly a year ago in Nature, and we now have a follow-up paper in bioRxiv, which was just recently accepted, looking at vasculature dysregulation in AD across different brain regions and across different genetic associations with Alzheimer's. And we're now looking into single-cell DNA accessibility and the correlated changes that we see between DNA accessibility and RNA expression, enabling us to start predicting upstream regulators for these disorders that we can now start manipulating systematically. We had previously reported on SPI1, we're now finding BCL11A and B, and we are also using that to localize genetic variants very specifically to not just neuronal pathways for psychiatric disorders, but very specifically microglia, as we had postulated back in 2015 for Alzheimer's disease. Very excitedly, we can look at not just the changes in expression, but also changes in the sequence of individual genes using these full transcriptome sequencing, such as SmartSeq. We can basically start asking what is the number of mutations accumulated by each individual cell. So here we're clustering cells based on their transcriptome, but coloring cells based on their mutational burden. And what we see is that Alzheimer's individuals show an increased mutational burden, very specifically in these de-identified cells. These cells have basically lost their identity, and they are accumulating mutations in very specific pathways, which are associated with energy production, signaling, lipid metabolism, and transport. Those individuals that show these de-identified cells are in fact also showing this dramatic phenomenon of epigenome erosion. So early AD individuals have no change relative to non-AD, but late AD individuals show this dramatic closing of traditionally open regions such as promoters and enhancers, and this opening of traditionally closed regions such as her chromatin and quiescent regions. And you can see here these dramatic alterations. So what we believe is happening is this breakdown through an accumulation of DNA damage and DNA double-stranded breaks associated with repair, but also memory formation, that is basically leading to ultimately this breakdown of the overall nuclear organization, detachment from the lamin regions, and reorganization in high C regions in data that I'm not showing here, where you can basically see that non-AD and early AD individuals are much different, but late AD individuals very specifically show this shift in these de-identified and dysregulated cells. We're now looking across multiple phenotypes to start connecting these multiple different lines of evidence. And in particular, what we're finding is that when you look at these 427 individuals that we've profiled at single cell resolution Alzheimer's disease, you can discover 1,600 genes that are differentially expressed, and you can cluster these across modules and hallmarks of disease. In the same way that we can think about the hallmarks of cancer, we can think about the hallmarks of Alzheimer's disease. And what's very exciting here is that these hallmarks are in fact correlated with specific biological processes, enabling us to start thinking about how to target each of these hallmarks, and specific phenotypic variables of these individuals, that the transcriptional signatures of these individuals in fact correlate with phenotypic signatures of these individuals across cognition, education, pathology, Braak stage, cognitive diagnosis. And we can now group these hallmarks of disease to start defining subgroups of patient subtypes. And that allows us to now find some subtypes that are very depleted for late stage AD, other subtypes that are very enriched for late stage AD. And what we're seeing is that these are associated with distinct hallmarks, either early versus late, male versus female, old versus young, and these diversity of phenotypes. That's very exciting because we can now start predicting these hallmarks transcriptionally. We can basically, and also genetically, we can look at the genetic variants associated with each individual that allow us to predict global transcriptional changes across these 1,600 genes, and then the specific class of individuals that they belong in.
Manolis, we're going to start losing people in a couple of minutes.
I'm so sorry. I'm going to wrap up in 10 seconds. So basically, we also looked at longitudinal profiling, looked at cancer and the microenvironment, looked at changes associated with metabolism, and then very excitedly, we're now able to reprogram these cells and start activating, repressing, editing, or knocking down. We've also developed these methods of carrying out millions of experiments in a single assay. And then we're also profiling individual changes associated with the brain. And then lastly, designing these embedding models that allow us to jointly project chemical structure, protein structure, discover these domains that are altered, and start understanding the organization of the single cell data in sort of a more geometric approach in collaboration with Marika Zitnik, and then designing these molecules and synthesizing them with Brown Petalute. So overall, I think that there's a very big promise for personalized medicine. We really need a multidisciplinary approach to sort of connect all of that. I'm thrilled to be collaborating with this incredible set of both computational and experimental individuals. And then I'll stop there and take any questions. Thank you.
Wow, thank you. What a tour de force. I think you did so well to cover all this in 50 minutes. It would have taken me 5 hours, probably, if not longer. So I'll open the floor for questions, particularly for those who need to go shortly, and then we can continue after. Fabian.
Thanks so much, Ile. Thanks, Manolis. This was impressive. That was dense, and I always learn something new, even though sort of I know your general agenda. So I also learn you can listen to talk multiple times and sort of pick up stuff. Yeah, I was really impressed by the whole Alzheimer story that sort of starts with genetics and the omics, and then goes toward single cell. And I'm always excited and interested in thinking about how can we use our tools that the field is developing in single cell level to then also contextualize with genetics that you've been so beautifully going through in this example. So my main question would be, what in your opinion is the main use of single cell resolution now for doing all kinds of multi-omics, maybe also spatial, as you showed below? Do you think it's typically enough to aggregate on a, say, subcell type or so position and then do this essentially sort of selection, like a fax thing, so to speak, then going onto mini batches, pseudobulk, and then doing the association? Or do you think there's also something that we can do sort of with transitions or something more fancy with networks or whatever?
Well, the answer is, as you would expect, it depends on the application. So for example, here, being able to sort of look at this gradient between these well-defined cells and these de-identified cells made a very big difference. If we had simply thought about oligodendrocytes, we wouldn't have found it. Moreover, we had to actually go back and
I was thinking about oligodendrocytes. I was thinking that has to be the example, right? Because that's like the continuous.
Yeah, exactly. And we were able to rescue cells that would otherwise be, sort of thrown away. So I think the power of the single cell data is that we truly have the ability to sort of look at multiple scales of resolution. You can look at the pseudobulk level for major cell types. You can look at the pseudobulk level or pseudocell sorted level for, smaller cell types. Or you can look at sort of the specific regions of alteration. So here, for example, I'm pointing out that, there's specific, like with these red arrows, there's specific places where you see more cells from AD plus B individuals. If we had thought of astrocytes as simply a uniform cluster, we wouldn't be able to see that. So you could even do that with graphs, basically showing the relationship between those, and then looking at subgraphs of 500 cells. And then seeing how these subgraphs are in fact changing their positioning between risk and, sort of healthy and risk individuals, or affected and non-affected individuals. So I think it's very powerful to be able to sort of look at these multiple resolutions simultaneously. And that would be my answer, that basically for some things, you can just look at the global changes. And maybe that's a good place to start. You just look at sort of, 30 subtypes of cells, and that's it. And then after you have your signatures, you can then see where are the signatures located in different subsets of cells. I hope that helps.
The relative frequency, I can see the one thing that I think you have not shown, or maybe I missed it, was, we also sort of start building more of these reference atlases. So you can also use the power of single cell to sort of more efficiently map, because you have all these micro variations and so on in the representation learning. Have you been thinking about that, maybe for faster annotation of sort of enriching with additional omics levels, or sort of just integrating multiple data sets?
Yeah, I mean, that's something that we've done time and again. We basically initially redefine the map for every data set. That's what we did for chromatin state. Every data set, we would just redefine them. And then eventually we just said, let's just fix those and just go after it. And I think that's sort of a little bit of where we're heading to now. I mean, for every new data set, we kind of recluster. And then there's just so much information now that we can kind of reuse these annotations. But at some point, instead of just reclustering, we might just say, let's annotate every cell de novo. And a lot of these
exactly just sort of really map each one, without just cell to transfer.
Yeah, and a lot of these language models, these deep learning models, these pre-trained models allow us to now just like forget all that and just say, okay, let's go in and just predict the cell type and tell us what accuracy you're predicting with. And then, maybe even do the control and correction, at the individual level without sort of having to think about groups and batches. So I think there's a huge power ahead for these pre-trained models.
Strongly agree. I'm gonna put like one reference papers that we've been doing in this direction in the chat.
Perfect. Thank you.
One really nice presentation. I need to run because I have my next thing, but that was really enjoyable. Thank you.
So I see that there's a Slido link here. Let me see.
Yeah. I have a question, Manolis. Can you tell me a bit more, or tell us a bit more about this? What did you call transcriptional resilience?
Yeah.
That you saw in women for Alzheimer's.
Yeah, so there's a lot of places where we find this. So let me just go back to sort of what I mentioned earlier. So basically here you basically have some individuals which are so here they're schizophrenia individuals, but they show control-like cells. Is this because they're transcriptionally resilient to schizophrenia? Or is it that these individuals here are basically phenotypically resilient? And even for Alzheimer's, we basically, let me see, just 1 second. I'm not going to find it now.
So, but transcriptionally resilient, you mean that despite having the molecular profile that would normally cluster them with other individuals of the same disease, they in fact do not express that phenotype?
That's basically, again, this is one interpretation of the data, but there's multiple interpretations of the data. But that would be one of them. And then what I showed earlier for this very first paper of Alzheimer's, we basically see that female individuals here were selected to be in our cohort to have exactly the same phenotypic severity. The female individuals here are not phenotypically more severe, and yet they're transcriptionally more severe. So this would be a case of phenotypic resilience for these female individuals. So it is possible that they're able to tolerate much more transcriptional dysregulation without actually showing it at the phenotypic level. So this is sort of, one interpretation of the data. But again, I think what we need to do as a field is basically embrace that we need to perhaps start redefining phenotypic clusters through a combination of both transcriptional and phenotypic variables. And rather than just simply saying, oh, the phenotype is correct, anything in the transcription must be wrong. I think that sort of a joint relearning of these phenotypic subclusters through combined analysis with transcriptome would be, perhaps the right approach.
Thanks. I see on Slido that Matthias had a question. Matthias, do you want to unmute?
Yeah. Hi, Manolis. Thanks for the great talk. And I mean, yeah, really overwhelming. Just pick out one analysis that I found quite interesting, which was the methylation QTLs that you showed, and how you used this genetically predicted methylation to link it to the phenotype. And I was wondering, I mean, the methylation is also age dependent, right? So how do you use this information? Does that add you something, or can you learn something more?
Basically, in this particular case, we have, let's see, go back to that slide. I had it a second ago. There we go. So basically, in this particular case, we're focusing on just, quote unquote, just 50,000 loci, which are genetically predictable. There are, something like 250,000 methylation probes. only about a quarter or a fifth of them is in fact genetically predictable. So the vast majority of them are not. And that basically says that, yes, indeed, many of them are just simply age-driven or differentiation-driven, or maybe disease-driven. But here, what we're saying is that we can't do that across the whole genome. But for the subset that are genetically predicted, then we can start building these maps of causality and sort of mediation. I hope that helps. But
I was just wondering because, I mean, you could have a genetic and an age effect, and because Alzheimer is sort of a late disease, the question was like, could you, for example, make this, use this extra information on age, or maybe a gene by age interaction also to make this more precise?
Yeah. No, I agree with you completely. One of the things that we're thinking a lot about is, can we use now this imputed methylation and look at the difference of observed methylation? And that observed methylation is more likely to be environmentally driven. So you can, right now we focused on the genetic component, but we can focus specifically on the non-genetic component and then start correlating that with environmental variables. And there's a lot of work right now seeking to systematically quantify environmental factors. And I think this is one of the most exciting directions. I wanted to go back briefly, the slide that I was trying to find, I haven't found the actual slide, but basically here, we're looking at AD biomarkers in the blood, and we're classifying AD versus control. And you see this amazing group here of, AD with a sprinkling of some controls, and this amazing group of control with a sprinkling of some AD. The question now is, what distinguishes these AD versus clean control, dirty AD versus clean control, dirty control versus clean AD, and so on and so forth. So basically, there's a lot more to be learned in the sort of combined transcriptomic and phenotypic classification of individuals.
It's very cool.
I saw one question on Slido about methylation QTL analysis, the link between genetically predicted methylation phenotype is cool. Methylation is also, oh, that was you, Matthias.
That's Matthias, yes. Then we have to what extent do you think new genes discovery will play a role in complex disease genomics?
I don't think it's going to be that big. Basically, we are kind of reaching the tail end of the gene discovery phase, I think, for the human genome. We have immense power now with, more than 200 mammals, and we're able to sort of discover very, very short exons. It's going to be very rare to discover new protein coding genes. It's possible that new RNA genes are there, and these are much more difficult to analyze. They have much lower expression patterns, and there's going to be challenges associated with that. And then how effective are machine learning models in discovering new genes in the genome? So we haven't yet done sort of true machine learning models, if you wish. I mean, what we've done is these evolutionary signatures that look at every column of the alignment and then predicting from that column, just purely, I mean, I don't know if you want to call it machine learning or statistics, but basically we're looking at frequencies where we basically have this generative model that basically looks at how likely is it that this column of the alignment interpreted through the phylogenetic tree relating the species was generated by a protein coding model or a non-coding model. And we train these empirically through bootstrapping from thousands of locations in the genome that, or I should say billions of letters in the genome that are aligned in coding versus non-coding regions. And that gives us a 64 by 64 matrix across the tree length parameters to basically train this model. And that's how we get these incredibly precise annotations. So I don't know how much language models or sort of these deep learning approaches can improve on this, but I'll be very excited to see basically if we can sort of take a protein structure prediction model such as AlphaFold and run it at the same time as we're running these sort of comparative genomic models. Maybe this will improve the accuracy, but we're already doing incredibly well. Okay.
Very, very cool. Thank you so much, Manolis. I think that has given us a lot of food for thought. And thanks for making a link to your slides available. I'm sure lots of us will go through and just look at the multiple very interesting graphs again.
Thank you very, very much. Again, I want to thank these unbelievable set of individuals. I try to acknowledge each person by a picture, right on the slide, as you could say. And then
Yeah.
there's a lot of sort of links to additional information. But again, I'm just so happy to be working with these unbelievable people. And if folks are looking for postdocs, we're always hiring. Basically, we're looking for talented people. If people want to spend some time in the United States, you know, I hear that there's less cyber attacks here. Anyway, we'd be delighted if folks want to apply. So please.
I would be very happy, Manolis, to see you back in Munich in the next
know,
few
I can't
months.
wait.
It'd be lovely.
So this is our website, compile.mit.edu. Please, you know, do reach out.
Very cool. Great. Have a lovely day. Bye-bye. Bye, everyone.
Bye.